All right. We are going to go over Waldenstrom's macroglobulinemia, also known as WM. The name is so long because the person who discovered it named it after himself. So that's what we're stuck with. And the reason we decided to study this is because one of our group members actually has a family member who has this condition. It accounts for 2% of hematologic cancers and affects approximately 1,500 Americans a year. And it is a cancer of the lymph nodes. So IgM is the protein that we decided to research and dig deeper into. And 75% of our immune cells are actually IgG, different from IgM. And IgM is a protein that is found as a part of the 25% of the other immune cells. And the reason, we'll go into more about the structure of the protein later. And so it is the first antibody to appear when exposed to an antigen. And that's really interesting because even though it is found in lower quantities in the bloodstream, you have about 10 active sites that can bind to the antigen as opposed to IgG, which only has 2. And so it has 5 times the amount of binding sites than the antibody that is found in majority in your bloodstream. And so the cancer, though, occurs when IgM is produced in large quantities, which will then cause hyperviscosity. And so hyperviscosity syndrome is one of the most common complications with WM. And the reason it causes so many issues is because the thick blood is very difficult to circulate through the heart, and you have an increased likelihood of bleeding throughout the disease progression. Here we see the years of treatment and survival rates. Down here at the very bottom, you see that the longest that anyone's survived from this research has been 15 years, and that goes with treatment. The type of treatment that we're looking at that is really cutting edge and that we're familiar with most because Spencer's dad uses it is Imbruvica treatment. And that treatment is really cool. First of all, because it targets cancer cells only. And ibrutinib is the drug that inhibits protein kinases of those cancer cells so that it will either stop disease progression or cause death of cancer cells. And then we'll get later into the hydropathy. And so that's what this will show. The hydropathy, basically, what you can get from here is that you have a lot of polar groups and a lot of nonpolar groups. And with an immune cell, that's what you'd expect to see throughout the entire amino acid peptide sequence.
Getting into the biochemistry, Joe touched on a lot of the aspects of this protein molecule. As you can see, it's a very large protein, and it takes up a lot of space. This immunoglobulin M is, as Joe mentioned, it's the first responder antibody in our immune system. The disease Waldenstrom's was identified a long time ago, but it was has become a lot more better understood in recent years, particularly through next generation sequencing, which identified a mutation in a driver gene in the hematologic B-cell malignancies. So essentially, they identified a missense mutation at a leucine at position 265 to proline. hence the name MYD88L265P. And this mutation is found in 90% of the Waldenstrom's cases, which is very interesting. As you can see, the protein is very large, and so when the uncontrolled growth happens in the body, this immunoglobulin eventually starts taking the place of red blood cells and crowding them out, which is the direct cause of the hyperviscosity and the other side effects that do occur. As you can see, it does have the 10 binding sites in the blue regions around the periphery of the protein. And then from there, we did talk about the really cutting edge therapy, targeted therapy for the B cell signaling pathways, which is really interesting, which is the Imbruvica drug, how that works. But we just did want to mention some of the other treatment areas that are being utilized in trying to conquer this uncontrolled protein growth. As you can see from the list, there's a number of them: chemotherapy, corticosteroids, biological therapy with monoclonal antibodies, immunomodulatory drugs, and proteasome inhibitors. Here we have a graphic that shows the potential therapeutic pathways that are available due to the actual biology of the disease of Waldenstrom's. You can see right there in the center, you've got the ibrutinib, which is a BTK, so kinase inhibitor, and then a number of other treatments that are being either tested or run through clinical trials, essentially for creation of effective drug therapies. For example, you've got the idelalisib, which is a medication that's used to treat blood cancers. And this substance acts as a phosphoinositide 3 kinase inhibitor. So it blocks basically the isoform of the enzyme in the phosphoinositide 3 kinase. And there's many other opportunities for research and for development of drug therapies. Lastly, we wanted to talk about another interesting therapy, which is the drug of rituximab. And it's a monoclonal antibody that attaches to an antigen called CD20, as you can see in the graphic, that's normally found on B cells. And it just causes cell death in the cancerous cells. Often, something of note is that these treatments are used not just alone, but together with other chemotherapies or other cancer drugs to treat the disease and control the protein.
So like other diseases, a very important aspect is research in order to help those who have been diagnosed and to help their families to be able to cope with this or to be searching for a cure. So there are 2 different research foundations which are known that conduct this research for the Waldenstrom's macroglobulinemia. The first is the Lymphoma Research Foundation, and second is the IWMF. And they fund the research for this that goes towards this disease. The IWMF is a small nonprofit foundation. Its resources are limited, and its sole donations are from members in order to fund the research. The goal of this foundation is to advance the knowledge of the pathophysiological basis for the disease in hopes of finding a cure. Each year, the IWMF accepts 2 to 4 new research proposals funded and carried out each year. Each year, these 2 to 4 new research proposals will go from any time to a year to 3 years. At $200,000 per study per year is what the IWMF gives to those proposals. With the process of research, the International Waldenstrom's Macroglobulinemia Foundation has funded research and uses these following processes. First, they have researched the signaling of the WM, determining the pathways that the cells use for communication. Second, they look at the immunology and the immunotherapy, looking at the human body and the immune system to see if it can better fight off this disease. Third is tumor microenvironment. How does the tumor/bone marrow environment influence these cells? And the omics. What can we learn further about the genomics, the epigenomics, and the mutations that cause this disease? Current research that is being carried out by this and funded by the IWMF this year are targeting the MYD88 and the L265P driver mutation in Waldenstrom's macroglobulinemia, like we discussed earlier, and that is being studied at Baylor College of Medicine. The Dana-Farber Cancer Institute is using CRISPR-based functional determination of the WM cells, testing strategies to overcome resistance. And the University of Amsterdam is finding realistic therapies for Waldenstrom's macroglobulinemia using kinome-centered loss of adhesion along with synthetic lethality screens.
So in summary, Waldenstrom's macroglobulinemia is a rare type of blood cancer that, due to a mutation, causes white blood cells to produce an abnormal quantity of the immunoglobulin known as IgM, which in large concentrations makes the blood more viscous, makes it harder to flow, and causes heart problems. With treatment, expected lifespan is between 10 to 15 years, which is slow for a cancer, but can be improved. Like we said, one of the ways of treating this disease is by treating with Imbruvica, which is notable for interfering with the protein kinases that allow the cell to produce such large quantities of IgM. Because it is a rare disease, there is not nearly as much research as there should be, but one of the things that is currently being researched that will be of great help is the potential through CRISPR to isolate the single mutation that prevents the cells from turning off IgM synthesis and effectively eliminating the mutation and the cancer.